Welcome back to Chemistry Laboratory. This is Dr. Rajshekar Medidi, Chemistry Department, University of Kabul. Today we'll discuss about the very important and advanced experiment that is paper chromatography and thin layer chromatography from the practical point of view. Our students from Kabul University, they have been performing the experimentation physically, visually, practically in the laboratory. So today we'll discuss about the paper chromatography, how the separation takes place, and the thin layer chromatography, how the process is applicable in this video. Chromatography basically may be defined as a separation technique that is to separate, identify, analyze, and quantify the components present in the mixture that is distributed between the stationary phase and the mobile phase. Basically, chromatography is a separation technique that is used to separate, identify, quantify, and analyze the components present in the mixture. These components are separated by means of distribution ratios between the stationary phase and the mobile phase. Mainly, the separation is depends upon the distribution factors between the stationary phase and the mobile phase. We will try to understand the paper chromatography and thin layer chromatography together because the method and the separation technique is the same for both the experiments. So, in the case of paper chromatography, we are using the paper as the stationary phase, and the mobile phase can be used as different solvents. But whereas in the case of thin layer chromatography, we are using the thin layer coated with the silica on the surface of the plate, can be used as a thin layer plate for making the separation. So in the case of paper chromatography, we can use the cellulose paper of different dimensions we can use based on the requirement, and the paper is used as the stationary phase. But whereas in the case of thin layer chromatography, we are using the coated silica plate as the stationary phase. The solvents we are used commonly for both the experiments as same, that is butanol, N-butanol, and acetic acid and water can be used in the ratio of 4 is to 1 is to 5, whereas we can prepare the different volume of the solvent as the mobile phase, maybe around 250 ml for both the experimentations. We can take 100 ml mobile phase for the paper chromatography and the remaining 150 ml mobile phase for the thin layer chromatography. Before arranging the paper inside the jar, we have to mark the line on the paper with the pencil so that we can take the three samples like amino acids. For the samples preparation, we can use the same samples for the two experiments for TLC and paper also. We can take the approximately 0.50 microgram of the sample, and we can add one drop of water to make it as a slurry or a paste something. Like we can take that by means of capillary tube, we can apply on the spot of the paper which is to be marked as a sample spot. After making the arrangement of sample spotting, we can keep the paper, as you know already about the experimentation, we can keep in the jar containing the solvent, that is N-butanol, water and acetic acid. So because of the capillary action, the solvent will be rising up with a different ratios, so that we can see the three samples will be separated slowly when the mobile phase is moving up by means of the capillary action. Same is the case with the TLC also. We can take the three samples in the different arrangement. Maybe the some bowl is used for the plate to be kept properly, and we can make the marking on the TLC also in the same way with the pencil. We can spot the three spots of the samples as like in the same procedure for the paper chromatography. Then we can see the mobile phase will be slowly rising up because of the capillary actions. So because the two experimentations, maybe it is different, paper and TLC, but the samples are the same, we can get the same similar type of separations in this case. So after the when the solvent is moving up as a solvent front, we can make the margin up to where the solvent has been raised up. Then we can measure the spots by making the marking with the pencil, so that we can measure the centimeters, how much distance the each sample is traveled. So let us perform practically, visually by means of the students. Now the students are there, post graduation students, MSc chemistry, advanced analytical chemistry. They have been performed these two experiments visually, practically in the laboratory. Let us see the experiment now practically. Now let us see the preparation of the mobile phase, that is 4 is to 1 is to 5. N-butanol 4 parts, 1 part acetic acid and 5 parts water. So we are preparing 250 ml of mobile phase for both the experiments. So 100 ml of N-butanol, 25 ml of acetic acid, and 125 ml of water is required. So we are measuring by means of the measuring cylinder and preparing the mobile phases for both the experiments. You can see. So after preparing the mobile phase, that is the setup for the paper chromatography with the jar. You can see, and we will use the chemical called as ninhydrin. That is very important. That is there at the back side of the rack. Yeah. So we can use that ninhydrin for identifying the identification of the amino acids. So when we spray the dried paper after finishing the experiment and a dried thin layer after finishing the experiment, we can spray the ninhydrin on the paper and the TLC so that after drying, the amino acids reacts with the ninhydrin to produce the purple colored spots. So now we can see the spotting of the thin layer chromatography by the students. They are making the margin with the scale by means of pencil. So they are measuring the distance of each sample because three samples are there. So they are making the equidistance between the three. So they are marking the spots now. Sample 1, sample 2, we can see, and sample 3 also they will spot. So this is about the spot of by making the pencil. They made like that. Now this is the paper chromatography. So they have taken the margin for the paper also, so that they will keep the paper inside the jar. So they are marking the same way. And now the TLC, they are spotting the samples. Already sample is dissolved. We can take 0.50 microgram of sample with 1 or 2 drops of water. We can keep the spots. So this is spotting on the paper. Both experiments are going on simultaneously. Both, that is paper and thin layer also. So 3 samples for each. So this is the paper chromatography spotting. 3 samples, and thin layer also 3 samples we can see. So after drying, that means after completion of the experiment, we can spray the ninhydrin. This is the bottle they are used for spraying the ninhydrin on the dried paper. That is paper chromatography. The experiment is over and it is dried. After drying, we can spray uniformly so that again after spraying it will be dried, so that we will get the purple colored spots developed and it will be visible clearly. So these are the 3 different samples we have taken by means of watch glass with the tube we are used, and for both the experiments capillary tubes are used for both the experiments to get the spots. So now after this is the thin layer chromatography setup with the tub taken 150 ml of mobile phase, and it is kept immersed into the bottle, and this is the jar with the paper chromatography immersed inside the with the mobile phase. So they have done the experiment like this. So after finishing the spraying, after drying, we get the spots like this. 3 different samples with different spotting is observed practically. So they will mark now on this, and they will measure the centimeters traveled by the spots from the bottom to the solvent front. So this is TLC spotting, 3 samples. So since both are the same samples, so we got the same type of separation for this both. So after this, we'll make the calculations. So after completing the experiment, we got the results like this for the paper chromatography and the TLC. We can see. Now, based on these experimental results, we can find the calculations. That is, for experiment 1, paper chromatography, RF value, that is distance moved by the sample by the distance moved by the solvent front. So we got the values for the 3 samples. Sample 1, lysine, sample 2, cysteine, and sample 3, L-alanine we can take. So the values obtained is we can see, And for experiment number 2, TLC, these are the values that is obtained for the 3 samples, and the calculations we can see. We can find from experiment 2, that is first sample is 0.13 RF value, second sample 0.06, third sample 0.4. So we can perform the experimentation like this with all the calculations. Thank you.